Hello, I'm Robert Walker, and this is about the paper that I'm about to submit to an astrobiology journal sometime, probably next spring. And it's, as you can see, it's quite startling in its conclusion. It says that NASA will not be able to complete the process of returning the Mars samples by their timeline of 2031. They've already launched a mission to Mars, Perseverance, to collect these samples, and then there are going to be other missions by ESA and to go there and pick them up and return them to Earth. And they haven't yet started the legal process. So this is a multidisciplinary paper, and I want to talk about it first. This is a presentation in hope that experts will have a listen and have a read of the paper and see if they can double check the things that I've said. It seems pretty clear. So let me explain why it will take so long. If you'd read some of the reviews, and I recommend Oren et al., an excellent review of the literature, and they say that if you look at their review, it seems just about possible that everything could be completed by 2031 or 2, maybe delayed a few years to 2033 or something. But so if I have a look down at their, I've done some extracts from my paper here. So this is the outline timeline, and it's 8 to 9 years for the legal process, including preparing for it, which hasn't yet started, and then 11 years for the build. And the facility that would need to be built, an estimate cost about $500 million. But you would think from that if we start the build right away and we start the legal process right away in 2020, we could complete it all by 2031 and be ready to receive the samples. And given that it's not going to start in 2020, then it would probably be delayed by a year or two, but it would seem to be possible. However, there are a couple of issues with this that I have identified, which doesn't seem to be noticed in the planetary protection literature so far. First of all, we don't have the technology yet for the build. And so I'm going to explain that. And secondly, NASA do not require an enterprise, I've got it written down what they call it, an enterprise, is it mentioned there? Yes, an enterprise architecture project review and an enterprise architecture service review before they approve the budget and before they start the build. Even if you've got some donors, if you've got Elon Musk or something, gave them $500 million so they don't need their budget, they still wouldn't start the build without doing these things. So then the question is, how soon can they know what it is they have to build? And I argue that they won't know until the completion of the legal process. So that means the two lines have to be one after another rather than together. So that takes us to 2040, starting in 2021. It's very hard to see how it could be completed before 2040, before they can return the sample to Earth. So that is the nub of my paper. So that's the first thing I want you to, I'll explain that in a bit more detail. And please, any experts out there, engineers, legal experts, astrobiologists, please, can you double check? I've talked to people and I can't, do you see anything wrong with my conclusions here? Is there anything that I've missed out? I've talked to several people and they can't find any mistakes in it. I've been encouraged to, I've been writing articles about the planetary protection for my blog posts on a what you call it, for public, for anyone to read. And I've been encouraged by an astrobiologist friend to write this up as a proper paper for submission to an astrobiology journal. So I've done that, and it's nearly completed, but before actually submitting it, I wish to do like the equivalent of giving a seminar about my research and to get feedback from experts. And by doing it on YouTube, then I can reach a large number of people if they're interested. And I will share this with the Virtual Astrobiology Society and with friends via email and that sort of thing. So, now, why do I say that it's not technologically possible at present? So, and I will also argue that the European, the space, the sample return facility, the sample return studies, they have a remit to find a safe way to return a sample from Mars to Earth. However, in the legal process, the remit will be different. It will ask, is there a safe way to return a sample from Mars to Earth? And this actually, you see the confrontation if you look at the European Space Foundation Mars sample return study in 2012. They have a section where they examine what version of the precautionary principle we should use. This is a principle that's been developed over the last few decades to do with what happens if you have a possibility of large-scale adverse effects. And even when you don't have the sufficient knowledge to be able to work out what the effects will be. And the precautionary principle is that that the proponents of the activity are the ones that have to do the research to show it is safe, rather than the ones who are examining the process have to do that research to prove it is unsafe. They have to prove it's safe. And the various versions of the precautionary principle, the prohibitory version would say that if you can't prove it's safe, you can't return it. And then the best available technology version says that you find the best available technology to return it. And that's what's necessary to show that it's okay to return it. And the European Space Foundation study, they examined this, and they said, given their remit, that you have to return it safely, and then looking at the precautionary principle, and if you are not permitted to return the sample if there's appreciable risk of harm to the, even very small but appreciable risk of harm to Earth's biosphere, then you simply wouldn't be able to return it. So they said, as their conclusion, that you have to use the best available technology version of the precautionary principle. Now, when you go to a legal process, they don't have that same remit. So they wouldn't argue in that same way. And some people, and an example would be Carl Sagan. So if Carl Sagan was talking and advocating, and sadly he's died of course, but if he was still alive and he was talking to the legal process, he would argue that we cannot take a small but appreciable risk with 1 billion lives, even if it's very small. Carl Sagan argued that we can't do that. We just shouldn't do that. So, if the technology is the same as for the 2012 study, if the conclusion is the same, that we can't eliminate a small but appreciable risk, then the end result of the legal process would be that it's prohibited. So I'm not saying how likely this is, but there's certainly a legal possibility that the conclusion of the legal process is that it can't be returned. And so, given that background, there's no way that NASA is going to release the budget or approve the build until the legal process is completed, because they won't even know if they are permitted to build the building until the end of the legal process. So that's why I think it's pretty certain that they're not going to start build until at least 9 years from now. And if you look at all the ramifications of the legal process, it's extremely unlikely that it would be completed, I think, in as short as 9 years. So 2040 is like the earliest possibility. Again, if any of you are legal experts, if you're technology experts, please can you have a look at my paper, which I'll put in the video description. Is this correct, what I've just said? And for those and just make it absolutely clear, this is a preprint, it is not peer reviewed. And it's going to go through the process of peer review if it's accepted. And I imagine it'll be quite a complicated, difficult one to peer review. You'll need many experts in different areas of expertise to peer review it. That's unavoidable because of the nature of the subject. And I did consider whether doing the small papers one after another, but there's the timescale thing, because ESA is already starting work on the missions to go to Mars to pick it up. And by the time I publish 4 or 5 papers, and each gone through peer review, then they will already have sent their missions to Mars to pick it up, and the legal process will be going through, and this will be a great letdown, a decade from now. If what I'm saying is true, then I am not putting an obstacle in the way of a sample return. I am simply describing something that's going to be on Earth as we go through the legal process. The Mars sample return studies are not sufficiently thorough to predict the legal process. They haven't had, for instance, the World Health Organization is definitely going to be involved, in my view. That sort of expertise has not been used in these sample return studies. So, anyway, now I'm going to go down to, and the Owen et al. To mention that the World Health Organization is likely to get involved. So now, what is the technological problem? Why is it technologically difficult, even with that 1 in a million chance? So even with the European Space Foundation study, and they're saying we've got to use best available technology, still their requirements, which are not sufficient to completely 100% prevent any appreciable risk, still is beyond current technology. So let me explain that. So the 2009 study by National Research Council set a limit of 0.25 microns for released particles, based on their understanding of minimal size of microbes. In just 3 years, by 2012, that was reduced to between 10 and 50 nanometers. That's an order of magnitude, more than an order of magnitude reduction. So how did that happen? Depending on how you look at it, or a 5-fold reduction if you look at the 50 nanometer number. So the reason is of new discoveries between 2009 and 2012. First were the filterable bacteria that can go through a filter of 0.1 microns. And this is an actual, later in their study, we actually have photographs and micrographs, of electron micrographs of these. Sorry, I need to press that. How do I get rid of it? Anyway. There we are. So this is an electron micrograph of I've got extract presentation here. I can maximize it. So there we go. And that's an electron micrograph of a scanning. I export it as a PDF of a scanning electron micrograph of a bacterium that passed through a 100 nanometer filter. And there, that's 100 nanometers roughly measured. So their 0.25 microns just wasn't big enough, wasn't small enough to have assurance that Martian bacteria won't be able to get through it. And then they also consider gene transfer agents, and these with the minimum size of 30 nanometers, these transfer, so for instance, gene transfer agents transferred the capability to express carotene from fungus to ants. Now, that was known long ago, but the question was, what's the time scale for this? So they discovered this can happen, this was discovered in 2010 research, discovered this can happen overnight in seawater. So if a single gene transfer agent got released from the sample return, got into the sea, then by the next morning you could have microbes that have already taken up that gene transfer agent in the worst case. This was a transferred property of antimicrobial resistance in this particular case. It was transferred overnight. So as a result of considering it, so now we're going to a legal process. Legal process will not ignore the European Space Foundation study. Again, I'm just saying what is going to be examined. I'm not adding anything new here, except putting it all together and making connections that don't seem to be made before. So the minimum size is unacceptable if a single particle cannot be released at 0.05 microns, and at 0.01 microns, which is smaller than the smallest viruses and smallest GTAs, then at that level reviews are needed. It's potentially tolerable, the lowest achievable at reasonable costs. And that's the best available technology thing, where they say that even though there's a very small risk of appreciable harm, it's impossible to completely eliminate that, and so you have reviews of what's potentially tolerable. And they came to the conclusion that a 1 in a million chance of these severe outcomes is tolerable. And many people in legal processes will say that is not tolerable. The likes of Carl Sagan would say that's not tolerable. So now the SF study also said that given this very dramatic reduction in the size requirements from 250 nanometers to 50 nanometers / 10 nanometers in just 3 years, they say they say that based on their current knowledge it'd be at a slower pace, but you can't disregard the possibility of future discoveries of new agents, entities and mechanisms that may shatter our current understanding. And so it's recommended that it is reviewed, that the requirement is reviewed and reconsidered on a regular basis. Certainly by 2018, 2020, 8 years later, it needs review. And one of the, I have got this in extracts, I could have to go to the paper, but for one example, in the last 8 years, since 2012, there's been a great deal of research into RNA world life, attempts to start evolving minimal cells from scratch, and synthetic minimal living cells, and synthetic biology. There's been a lot of research on that in the last 8 years, which is good because it does need to be reviewed. And in particular, this goes back to 1999, theoretically, RNA world life has only RNA, no DNA. A hypothetical life that would be simpler, there must have been something simpler that existed before the modern life, which is far too complex. Because modern living cell, that 250 nanometer, or even that microbe that bacteria I showed there, this could not arise in one go from non-living chemicals, and just suddenly you've got a bacteria like that. That's impossible. So what happened in between? And so then the RNA world is a suggestion that what happened in between had just RNA without DNA, very complex process of DNA transcription and all that. They just had RNA, no messenger RNA, just RNA is the only, a single informational biopolymer, RNA. And there were no proteins. And you didn't have the huge ribosomes, which are the main thing that limits the size of very small bacteria. You had maybe ribozymes. And ribozymes are much smaller. They wouldn't need to make proteins, just enzymes. There might be 5 genes in total. The very early life forms obviously didn't have the number of genes we have now, in even the simplest microbacteria, and they may have started with as little as 5 genes. And before that, something that didn't reproduce exactly, with less complexity even than that. So if you look at that minimal life form, then they worked out that it could fit in to 14 nanometers in diameter. The equivalent of this bacteria could be only 14 nanometers in diameter. Now, the European Space Foundation, in their study, then they didn't think you needed to go down to that level. But with the research since then, that needs to be I think this needs to be re-evaluated. And it's not impossible that this unacceptable level goes down to 0.01 microns, and that this whole region, if I can move that, this whole region turns red. It's not impossible. So this needs to be done urgently. We don't know. We can't even start the legal process really properly until we've done the review of the size requirements. And that's not something you can go in a few days or weeks. So I don't know how much, a few months or a year or something, proper study of all this. So now, why is it that you get papers that seem to be saying that it's absolutely fine and no problem, we know how to build these facilities? Well, it seems to be due to a typo. So this is I know this is rather startling and surprising, but if you look at that, if you look at the actual, what they actually, I haven't got the screenshot there, but they, let's bring that up, and I'll bring up the screenshot properly in my article. I didn't put that into the extracts. So, oh, here we are, we're quite close to it, so it'll be down here right further up. I left out oh yes, it's be further down, just a minute, give me a minute. There we are, I forgot to put that in. So this is their recommendation 7. And you see it says 0.01 microns. That's rather confusing comma there, it'd be the French comma I think. But they seem to have misread that as 0.1 microns. And that is, so the probability of 0.01 microns or 10 nanometers should be less than 10, and that's for a single unsterilized particle. And they also misread that as being a per particle probability, that each particle that encounters the filter should have a 1 in a million chance of getting through. No, it's not that. There must be a 1 in a million chance of a single particle through the entire lifetime of the facility. And it's very difficult to achieve. And given how difficult that is to achieve, then that's why they said that it could be reviewed. But they didn't do that detailed review themselves. And then at least a single unsterilized particle larger than 0.05 microns is not acceptable under any circumstances. Again, they possibly, maybe the comma confused them, I don't know. I don't know why they didn't see that. But it's a typo, pretty clear. So, again, I mean, if you could see any other explanation, but it's pretty clear what the European Space Foundation study says. And they say 0.1 microns, the study says 0.01 microns. Seems pretty clear there was some glitch. Someone misread something. So now if you go to that, now you look at current technology. So the HEPA filters, they just don't go down to that level. They're not tested at that level, HEPA and ULPA filters. I look at the aerosols that they use to test aerosols, and they don't go down to that size. So it's not tested. And but this is the state of the art. So this is for COVID-19. And a virus for COVID-19 can be as small as 60 nanometers in diameter. Now, we don't have any filters that can filter out viruses at that size. When you look at the people who are working in intensive care units, they are totally protected by the filters they use. The reason is that the most droplets that contain the COVID-19 virus are much larger than this and can be stopped by their filters. And also, a single virus of COVID-19 can't infect you. It seems to require thousands of them, hundreds or thousands of them, and altogether. And so the filters they use in intensive care units do keep out sufficient of the virus so that they're safe. However, there's interest in designing a filter that is small enough to keep out even a single virus. Now, that technology doesn't exist yet. If you go back to these things, then obviously we could construct something like that. You could construct a plate with 0.05 micron. Remember, this is well beyond the optical limit of 0.2 microns, the typical optical resolution limit of a microscope, of the best microscopes, optical microscopes. You can't see these holes with optical microscopes. So, but we could have manufacturing processes that create 0.05 micron holes. If it's a single one, it's a little bit too big, then we've had it. So we've got to be a very careful process. But obviously we make these 0.1 micron holes, we could make 0.05 micron holes. The problem is making it into a usable filter that you can use in a biosafety cabinet or a biosecurity laboratory. And that's not been done. Nowhere near. So this is an attempt, and this is a 6-layer very complex filter, and it was able to keep out 88%. I've just got an extract of it. I'm not sure if I've got the 8% figure. It's 88%. They were able to keep out 88% at 50 nanometers. This is nowhere near. So if a particle, there's an 18% probability if a particle impacts the filter that it gets through, nearly 1 in 5. And that is nowhere near the requirement to keep out all particles of this size. So we don't have the technology. It just doesn't exist. We don't even have it. And this is not an actual commercial filter. This is an experimental design that is not commercially available. And we don't even have it as an experimental design that fulfills the requirements of the European Space Foundation study. So that's why I say that so then if you go back to this legal process, then we can't start this until we have the technology. If we don't have the technology, or at least an outline of how we're going to achieve the technology over a reasonable timescale, then there's no way it can pass through the legal process. And then you have to have the technology. You have to not just make the filters. You've got to explain how you can change the filters, how you can sterilize it and get it out without losing any of the without sterilize the filter in situ perhaps, and then remove it in some secure way that doesn't let anything from the sample come out. And filters typically have to be changed about once a year, or once a year or two, every year or two. So that's why I said, the enterprise service architecture and the service review, that would have to be completed. So they have to know how they're going to service these filters. They need to know what the filter is and how it's going to be serviced. Are you going to build this building in such a way as to be able to incorporate filters that have this capability and that can be serviced? And we're nowhere near that. So, and then if you look at the legal process, there are many things that they have to look at. They have to look at the environment, ocean, Great Lakes, escape of invasive species. Now, I just want to say quickly, now you might look at this and say, but there's no risk at all. You might be convinced. So I'll just talk about this briefly. If you have been a follower of Robert Zubrin, then he talks about how keeping microbes from Mars out of Earth is like keeping Canada geese out of the United States, and that needing to do it in a sample return is like putting the Canada geese in the boots of your car in order to transfer them from Canada to the United States. It's a very vivid analogy, but it doesn't apply to the Mars-Earth situation. I mean, obviously Canada geese can't fly from Mars to Earth, but he's just giving an analogy. But the most Earth microbes couldn't transfer from Mars to Earth. Most photosynthetic life on Earth couldn't transfer from Mars to Earth because of the shock of the ejection would kill them. And so the question is there any, and so what kind of life? And then if you look at the possible habitats, let's just go down to that. Let's see how we're doing. Go down to the possible habitats. So then I want to talk about this in a bit more detail. So these are Curiosity's measurements of indirectly detected surface binds. And this is before sunrise and sunset. Before sunrise, then just slightly after sunrise, then even at 0 centimeters, liquid forms on the sand dunes from the salt taking up liquid from the 100% humidity, especially in winter at night. And then through into the day, it's still liquid at 5 centimeters below the surface. And they haven't actually observed these binds, they can't sample them. Curiosity has no way to dig into the sand and try to find these binds, but they have found them indirectly through humidity measurements, and it's pretty conclusive evidence. These binds are not habitable for terrestrial life, because at night they're too cold, in the daytime they dry out. However, so there is no real appreciable forward contamination risk from single spores on the Curiosity rover for these, unless they already are inhabited by biofilms. So there's a possibility that they are already inhabited by Martian biofilms. Certainly, if ever humans went there, and with the amount of virus that we shed, of bacteria we shed, then we would create biofilms there, and the biofilms can trap the water from the night and retain it into the daytime, into times of the day when terrestrial life can reproduce. So, and so the warm enough temperatures so it would trap the water. So without a biofilm, it's not habitable. With a biofilm, this is an insight from Nielsen Benno, who's a top researcher into for instance, he wrote the big review paper on the possibility of liquid water on the surface of Mars. And he is of the view that there could be biofilms in these fine layers. That biofilms could make them habitable through to the daytime. And then, just to correct a kind of misconception some people have, there's a minus 20 degrees centigrade lower limit for reproduction of life that they use in the planet protection literature. This is not an absolute limit. It's some studies suggest it's possible to have very, very slow reproduction of life at below minus 20, but it could take centuries to thousands of years, tens of thousands of years for single reproduction, for single splitting of one cell into two. And so, because of those timescales, then they say we don't need to worry about that because it's not going to spread far on the Martian surface over human timescales. So when they say minus 20 degrees centigrade, for instance, for these plants, but they'll go right down to minus 40, I think. They go very cold. So if there is, even if terrestrial life got in there, then it might reproduce, but very, very slowly. So slow it's not a concern. But if you have a biofilm, then it can reproduce much faster. So it's really about the timing, about the reproduction. And so now Martian life, also the chemotropic agents on Mars, particularly the perchlorates, the sulfates, the chlorates, they help break hydrogen bonds, and they make it possible for metabolic processes to happen faster. So there's a question that is not answered about whether terrestrial microbes could take up some of the chemotropic agents from the Martian surface and use it to have faster metabolic reactions even at very low temperatures. And this is left as an unanswered question in the big review by Rommel et al. And then and I've tried to look, I can't find work since then that has resolved it yet. But it's certainly possible that Martian life could involve these chemotropic agents. And so it's entirely possible that two ways that Martian life could be surviving there. It could be surviving there as a biofilm, it could be surviving there using these chemotropic agents. And it could be there right through to the warmer parts of the day on the surface, not just at minus 5 degrees. And then we have the evidence from the, the Viking labeled release with the Miller et al.'s, Miller's discovery that the CO2 evolution is offset by 2 hours relative to the warmest temperatures in the day. And it's hard to explain that during chemical processes, and if the easiest way to understand that is that there's some form of life there in its circadian rhythms. And so there are increasing numbers of people, small still, but there are increasing numbers of people who think that it's possible that Viking did detect life in the 1970s with the Viking labelled release, and the various things that confused the other instruments, such as the perchlorates. So this all raises the possibility that maybe there is life on Mars that could be returned by the rover. So the question is, what do we do about all this? And the two possibilities, and one is that we I want to first explain why I also, so one is to sterilize the sample return, the other is to return it to somewhere safe in the Earth-Moon system. So but first I'll just talk a little bit more about why I think there's actually a very strong reason. This is new to me, and I can't find anything about this in the planetary protection literature. This particular thing that my suggestion that there could be a swan song Gaia homeostasis on Mars. So the usual, when you read the planetary protection literature, there's usually a lot of skepticism that there could still be life on Mars. The reason is that although it's barely habitable now, how could it have continued for 3 billion years? From early Mars, when it was very habitable, and may well have been life, but then 3 billion years, and so it would just need to be too dry for a while, and the life would go extinct. And then it maybe could be recolonized from below, but then that depends on there being life below the surface. And that's why many people think that we're not likely to find terrestrial Martian life on the surface. It's only likely to be deep down below the surface, because they're skeptical about it actually being able to colonize the surface fast enough to have got back there again after whenever it last went extinct on the surface, and or about it evolving fast enough to be there on the surface. However, I argue that what we have on Mars, so we have the idea of a swan song biosphere, and that's Jack O'Malley, let's look up swan song biosphere. Have I misspelled his name? That's a swan song. Sorry, but I've said a lot, so a little swan. Yeah, there we are. There's the swan song biosphere. If I go up a bit. Oh yes, maybe I do mention him here. So where we are. Oh, the swan song guy. Oh, swan song. I mean swan song. What's happening? What's happening here? Oh, I'm looking wrong page. So silly me. Yeah, here we go. Here we are. Suggestion of self-perpetuating swan song guy. So let's go to that. And that's, yeah, Jack O'Malley James introduced the idea of swan song biospheres. So a swan song biosphere is when, if Mars had terrestrial, so if you look at Earth's biosphere, then as the sun gradually warms up, and eventually it's going to get too hot for life here, we may well protect it with mirrors and actually moving Earth and things like that. But if you just look at the natural evolution of a terrestrial planet without a civilization on it, then if you go far into the future, it will get too hot for life. Gradually, the only life left would be at the top of high mountains like Mount Everest, their equivalent in the future. Then there would be life hanging on in caves in there. The sea's completely dried away, of course, long ago, and no ice caps. And then high mountains at the poles. Then there would be some life still remain. That's the swan song guy. That is a swan song biosphere. So the Martian biosphere. So the idea that all terrestrial planets are going to be teeming with life, then that's not necessarily true at all, because some of them could be in the swan song stage. If we're looking at exoplanets, and then life would be very hard to detect. Now, Mars could easily be in the swan song stage, but if you look back, it was habitable most habitable over 3 billion years ago. And so, how long would the swan song biosphere last for if there was life back then? And it would seem, on the face of it, that it wouldn't last for very long. Maybe a few 100 million years, maybe 1 billion years or something, 2 billion years, but eventually would be gone. However, what my new insight into this is, what if this swan song Gaia was self-perpetuating? So on Earth, then a swan song Gaia, then our Gaia on Earth makes Earth more habitable by taking carbon dioxide out of the atmosphere, and so cooling the Earth down. But now, if you go to Mars, taking carbon dioxide out of the atmosphere and making Mars cooler is not going to make Mars more habitable. And the many other cycles on Earth that help to keep it maintaining a homeostasis that is quite habitable for Earth. It's not necessarily optimal even on Earth. A warmer Earth would actually be more habitable in many ways, in terms of life generally. So it's not even clear that Earth, somewhat warmer, 10, 20 degrees warmer Earth would be much more habitable once life had adjusted to the warmer Earth. So it's not even clear that Earth is Earth's Gaia doesn't maintain Earth's biosphere in an optimal situation for life. It's slightly suboptimal. Well, the situation on Mars may well be very suboptimal, but a homeostasis all the same. And so that's my suggestion. And so if you look at it in the planetary protection literature, not in the Mars astrobiology literature, then there is people have, of course, remarked before, the remarkable coincidence that the Martian atmosphere is close to the triple point, where water vapor could be liquid, solid, or gas, and at that triple point for both pressure and temperature. And that's really quite remarkable. Why is it like that? And the very deepest places like the lowest part of Hell's crater is slightly above that triple point. We could have a liquid lasting for a little while there. And then the high mountains like Olympus Mons are well below the triple point, where it's very unlikely to get any liquid. But on average over the surface of Mars, it's just right to just barely be able to have these brines that Curiosity detected. So why is it so close to the triple point? Well, there has been a suggestion in the literature already that this could be due to abiotic photosynthesis. And things like carbonate, like the stalactites, stalagmites, carbonate formation, other processes like that involving liquid water on the surface of Mars. So it's just a very tiny little thing that I did. I said, well, what about biotic photosynthesis? Nobody seems to have raised that question. So what about biotic photosynthesis? And it seems quite possible that the same would happen. So if you had biology on Mars, so now we have to suppose there's an excess of carbon dioxide on the Martian surface being produced by the volcanoes, more than is needed to keep it warm enough for life. And from the Phoenix measurements in 2008, then the carbon dioxide in the atmosphere, the isotope ratios of carbon, shows that the carbon is exchanged with volcanoes. So volcanoes are producing carbon dioxide now, or at least not right now. We don't know of any active volcanoes, but in the last few million years, and it's also possible there are volcanic vents right now releasing carbon dioxide into the atmosphere. Mars is still geologically active, and the vents could be hidden by such things as for instance, the freezing conditions on the surface. And it's not impossible that there's actually carbon dioxide being released from the Mars surface right now. There's been many searches to try and find, and we haven't found the heat signatures of this. There's some spots that may just possibly be, but they don't seem to be. So Mars does seem to be it's certainly producing carbon dioxide over the million years timescale. We don't know if it's producing it continuously. And Mars, and then also from the same isotope evidence, then the oxygen in the carbon dioxide. So it's a very, very neat discovery from just the isotope ratio of carbon dioxide. The oxygen isotope ratio shows that the oxygen exchanged after being released from volcanoes with water on the surface of Mars. It's got the wrong isotope ratio to just be straight from the volcanoes. So this shows that there's at least chemical processes going on between the atmosphere and these brine layers that Curiosity discovered. And these brine layers could be very extensive over the entire equatorial region of Mars, wherever there are sand dunes. There could be a lot of brines on the surface of Mars. And so that leads to the idea that there could be photosynthesis happening in these brines. Remember, some of them are right at zero, right at the surface there. There could actually be photosynthesis happening in these layers, which by biofilms would retain it through. This is just modelled. This is not actual observations. And there could be liquid water being retained there, just below the surface, below a few millimeters of the dust would be enough to filter out the UV enough so they could have robust photosynthesis going on, which could take carbon dioxide out of the atmosphere and generate oxygen. So then you get if I go look at my presentation here, and so then we get this, the observation of seasonal variations of oxygen in Gale Crater, where you have in the summer more oxygen than expected and in winter less than expected. It's way below and way above the expectation. Now this could be through biochemical processes or it could be geological chemical things going on that they don't yet understand. But it could be life. So that could be the signature of the life that possibly Viking detected. So so I didn't quite explain. I didn't complete my line of thought for the Swan Song Gaia. So the idea is that the life has stayed on Mars all the way since. That whenever the carbon dioxide starts rising, then it takes carbon dioxide out of the atmosphere, and because of the constant surplus of carbon dioxide into the atmosphere, more than is needed to keep it warm enough for life, then it can remain barely habitable for billions of years. And that is the Swan Song Gaia hypothesis that I've put forward. That it's possible for life to be keeping conditions on Mars just barely habitable at the triple point for billions of years. And it would go over from time to time, but the life brings it back again. Now, there are two ways that can happen. It can also happen through the methanogens. So methanogens, if you suppose that there isn't quite enough carbon dioxide, but the methane from methanogens could bring it over, or the methane released, even from abiotic ancient methane being released to the surface, that could bring it over. And this is another process. We have the methanotrophs, and it was Cassie Conley who brought this to attention, that there are certain processes involving methanogens that create calcite, taking carbon dioxide out of the atmosphere and creating calcite. And if you had a source of methane, and above that some methanotrophs that are taking methanogens, generate methane, or methane from that was released, was sequestrated billions of years ago, and above that you have some methanotrophs that eat the methane, and then there's reactions. If it's a liquid layer, they can then use that to convert carbon dioxide into and the methane and the calcium in the rock, and there's some chemical process which forms calcite, which would block off the motion of methane up towards the surface. So this is again my own hypothesis there, that it's another possible feedback that there could be. And just like on Earth, there could be multiple reinforcing feedbacks that keep Mars in the swan song Gaia. So this is just a suggestion, and there may be other ones that could well be other ones going on. And so that way there would be that as soon as too much methane is warming the planet's surface up, then methanogens grow enough, there's enough liquid water, the water becomes liquid, and then the calcite forms, and then that blocks it off. And that would be self-limiting the amount of methane that goes to the surface. And that could, just intriguingly, that could explain the methane bursts, that it's some blocked off layer and then a little bit gets through. But that's very, very tangential, hypothetical, so I don't want to sort of push that. But the idea, it does seem reasonably feasible that there could be a swan song Gaia on Mars, maybe multiple processes going on, so that those are two processes, just like this on Earth, keeping it all at the same temperature of just barely habitable. And as soon as you get large amounts of liquid water, then it goes back. That would greatly increase the likelihood that there's present-day life on Mars, because there would be no opportunity for it to go extinct. It would never dry out enough. And you could work this out theoretically, if you could work out how much carbon dioxide has evolved. We can't even detect it yet, carbon dioxide. And so maybe the easiest way we find this is just by detecting the life. But that's a hypothesis. So we have these two possibilities, and then the question is, what do we do? So the two ways of doing it, we've got have I done the sterilising bit? No, I haven't done the sterilising bit, I don't think. So there are two approaches here. This is the simplest approach. We don't think there's any life in the sample. So now, if we look at past life, again, it's very unlikely that there is past life in the sample that is easily detectable, because of the huge influx of organics to Mars. Oh yes, I never explained about the Zubrin thing. So Robert Zubrin, he said that we got transfer from Mars to Earth of microbes. Now, if it is possible that this happened in the very early solar system, but these habitats I was talking about, with the just thin layers of brine on the surface, these would not be transferred to Earth. We don't have any samples of dust, Martian dust, or Martian brine salts on Earth. It's just never been transferred. It probably has never ever been transferred in the entire history of Mars, that particular habitat. And the reason is because the meteorite impacts the excavate material from at least 3 meters below the surface, and that's what gets to Earth. And the dust and salt, it wouldn't even be able to survive very easily, even if it was fired to Earth. It would just got, that's just going to go scattered off into the atmosphere, but it wouldn't even be sent this far. So we can't use the natural contamination standard. We can't say that Earth has received this life already, and so therefore it must be safe. And in particular, an example of what would be very unsafe would be if Mars has developed a mirror life, like a Cucuriciliopsis, which is a life on our Earth that could potentially survive on Mars. We think that there are lots of studies, including using Mars simulation chambers, that suggest that given a little bit of protection, just partial shade or a few millimeters or so of dust, then it could photosynthesize and survive on the surface of Mars. So now, if you had a mirror, a version of this, reflected, all this biology reflected in the mirror, and that was returned to Earth, then that is when you start saying this is not something that we can do safely. Simply, we just don't have the technology to safely return mirror life from Mars, if such exists, and if such has never been transferred here. And even if life has been transferred from Mars to Earth, the mirror life might not have been. Might have been both versions on Mars, normal life and mirror life, and maybe only normal life, through happenstance, has got to Earth. And the mirror life, and for instance the photosynthesis as well, the photosynthesis developed very late on Earth, and may have developed very late on Mars. It's not at all clear it even has photosynthesis. If it has, then it may have developed too late to be easily transferred to Earth, especially since it's very, very difficult for photosynthetic life to get here anyway. So Mars could easily have mirror photosynthetic life that has never got to Earth. And so then if you look at the people who research into synthetic biology, and there's a possibility that we actually have a roadmap for creating a mirror life bacterium, a mirror life microbe. And we have all the steps, but we can't put them together yet. But probably maybe sometime in the next decade or two, it's a very difficult process, we may be able to create mirror life in the laboratory. Very simple form of astrobiology that we know will work. Because the ordinary life works, if you reflect everything reflecting the mirror works, and they just need to put it together. Very, very difficult, because every cell that they use to try and as a starting point, everything is the other way around. But there are ways of substituting bit by bit to eventually get the entire mirror life cell out of that. Then researchers looking into the possibility of creating mirror life are very emphatic that we must not let this mirror life escape from the laboratory. And so they were designed in such a way that it can't survive outside the laboratory with some chemicals or something that it requires to survive. And so in the same way, we should not let mirror life from Mars come to Earth. Just under no circumstances should we permit that, unless we can totally contain it without any possibility of it being left out. If you look at how they map out how the future would go, if we got a mirror life form that couldn't, the thing is that a mirror life on Mars could quite possibly metabolize ordinary organics, because its biggest source of organics on Mars are the organics from comets and meteorites. And that's why it's so very, very difficult to detect past life on Mars. It's been degraded over if you look at an ancient delta, like the Jezero crater delta, then any past life there, then it has been, it's probably been warm for long periods of time, so it's lost its racemic signature. It's probably been degraded by the chlorates and perchlorates, sulfates and things. And it's had ionizing radiation. If it's come to the surface, then it's had ionizing radiation from the sun, and from cosmic radiation, and there's probably very little of it left. And then you have a huge influx of the comets and organics from comets, and we already have organics in the Martian meteorites that we're already studying on Earth. And you look at those organics, they can't prove whether it is formed by life or not. You can't tell the difference. So the astrobiologists say that the sample returned by Perseverance, if it's past life, it's not likely that it would give us more information, tell us a little bit more because it's from a particular location. It could maybe put together some more bits of the puzzle. It is not going to settle the question of whether that past life with organics was produced by life. They say very clearly this is only a technology demo for that. We have to search in situ with multiple biosignatures simultaneously to detect them, because you get imitation of biosignatures and by other processes for abiology, for instance the chiral signatures in meteorites. So we need multiple ways of detecting the organics as being life. We need to go to Mars, we need to search in situ in multiple different places. It's just not feasible to return enough samples from Mars to Earth to do it that way. We have to search in situ in multiple different places until eventually we found whatever the combination of events that managed to preserve past life to the present. Because most of the past life is expected not to be preserved, and even if the organics are preserved, they would not be recognizable as life. So, as far as the past life is concerned, then sterilizing a sample is no problem. It's not very likely at all that it even does return past life. although I do talk about some ways it could. So this is how we could sterilize it. For present day life, it would be recognizable as life if we sterilize it. So this is how we could sterilize it with an X-ray. Oh, this is going on quite long, so I'm going to, I think I'll divide this up. I've got 55 minutes. I'll do another video talking about how we can sterilize the sample for present day, sterilize the sample, we would recognize the present day life. And then because the sterilizing would only, so I'll just finish with the sterilizing, and then I'll talk about, in the next video I will talk about, I've got 5 minutes before we get to an hour, and in the next video I will talk about what we can do if we think there's life there. If we think there's a high probability of life. If we think there's a low probability of returning present day life, then the simplest thing to do is to sterilize the sample. And if you sterilize it with gamma rays, then have I got that down here, then the effect on rocks of gamma ray radiation, then it does not cause a significant amount of radioisotopes to decay for sterilizing dose of gamma rays. So you can still do the radioisotope dating. Geologists did this experiment and they found no effect on the radioisotope dates. There's very little effect on the actual the structure of the rock. It looks very, very much the same. The main difference they found, by this spacing was not affected in any way. The spectra, there was no evidence of dehydration. None of the crystals were destroyed. And the only thing that did happen, it did change the colour of quartz crystals from clear to deep brown and darkened, and they changed the colour of halite, salt crystals, to deep blue. So those are things that you can live with and allow for. Now, I suggest that we use X-rays. The reason is that X-rays, modern X-ray equipment, has become extremely low power and low mass. They use nanotechnology, nano emitters of X-rays. And so with the power of just watts, you could sterilize the sample. So you could sterilize the sample during the return journey. And this is why I think it's really quite important that we start thinking about this right now. Because European Space Foundation are going to be sending a satellite to support the Perseverance pretty soon, within a few years. And this satellite will then be used to return the sample from Mars to Earth. And then they're developing their rover and the sample container. Now, we could save hundreds of millions of dollars if we decide in advance that we're going to sterilize it, instead of only discovering halfway through the legal process. And then, having decided we're going to sterilize it, we then decide how we're going to do it, and we could build these X-ray emitters into the sample container. So because they're quite short range, they do the best way to do it. So, and the sample container would block out a small amount of the emitter. So indeed, materials, or you change the material of the sample container so it's transparent to X-rays. Either of those. At least you consider this in the design of a sample return container. And then you also would modify the satellite so that it then sterilizes it on the mission back from Mars to Earth. This would mean that by the time it got back to Mars, to Earth, and you would monitor, of course, to make sure what's going on, you have it trajectory biased away from Earth, just in case there's some major flaw and the X-ray doesn't work. And then by the time you got back to Earth, over the 6-month journey, or however long it takes, that is plenty long enough to completely sterilize the sample at just the right level, so that the biology is still recognizable, the rocks are still recognizable, and the rocks and the radioisotope still works. And then you can just do an air re-entry into the Earth's atmosphere. Very simple sample return. No need to capture it. You don't need to even, you don't even need to they have the idea of another mission going up there to collect it. But you wouldn't even need to do that. You could just do aero capture if you can manage to do it, to get it robust enough to resist aero capture. So that would be a huge saving. It saves sending that extra mission. It saves at least one mission. So this is something I think that should be considered and looked at. Can we sterilize it on the way back? So that's another thing I'd like, any experts here, please look at this. Am I missing anything? Is there something I should do? Anything to be corrected? Or this was this was about the oxygen, that they explained that the discovery that it depended on the opacity of the Martian dust storms, and if the dust storms, if it was very opaque, then you got less oxygen involved. It's a weak correlation that again suggested the possibility of life. It's rather harder to think of abiotic methods that could lead to a correlation like that. So anyway, I'm going to talk about all that later. I'll stop this now. If we've reached 1 hour, I'll talk about this in my second video, which might be a bit shorter. So the main thing is that for this first one, is that we can sterilize it. And I think this is the most likely thing that NASA is going to do. And I think by the time they go through this legal process, when they get to the end of it, then they will decide to do that. They could bypass the entire legal process and not have to do it at all if they decide now, in advance, that they're going to sterilize the sample. Because if it's sterilized, it's a very much simpler process. We can use the same process, we can just use the Outer Space Treaty. None of the environmental regulations of Earth comes into play. We can use the Outer Space Treaty and just return it in the same way we do returning samples from comets and asteroids. Because the sample is sterilized. So this is the simplest solution, and I think this is most likely what NASA will do, but it will likely only do it after having started the entire legal process and gone through the whole thing, and then done this review and all that, and then they discover that. But it might be that they've already decided to do this. They're working on it in consultation. But so if again, I've asked, what are NASA's plans, and everyone just says that they're going to return it, like this as an unsterilized sample. But if you know anything change in their plans, do let me know again in the comments. I just can't find anything about it. And so I think eventually they're going to sterilize the return. I think that the sooner we realize this, the faster, the better. However, there is a possibility that we can return life from Mars, and that could be a much more interesting mission for the astrobiologists. So that's what I'm going to talk about in the second half.